See, we already have some people online. That's good. Just want to remind you, people still show up online and don't do this. You have to email the words after class before 9 a.m. to ensure that you are actually paying attention, because anybody can log on and then go back to sleep. So send the words before 9 a.m., please. Morning. A little bit more than 1 minute left, so I'll do attendance. So Bangura is not here. Casebolt is here. Tristan is not here. Covington Griggs, not here. DeVere is not here. Dow is not here. Hughes is not here. Lebdy is not here. Little is not here. Mankins is here. Mathis is here. Raines is not here. Reed is not here. Elise is not here. Thompson is not here. Withrow is not here. Ian Dewey is not here, and Elkins is not here. And as I mentioned, if you're online, I'm just going to say you're not here, because until you send those attendance words at the end, technically you're not here. So make sure you send in those words before 9 a.m. If you're watching the video, so nobody who's watching at this very moment, if you're watching the video at a later date, as usual, you need to send in a screenshot with those words. When I say the words, you need to include a screenshot. And Lebdy is here. And it is 8, perfect timing. So we've done attendance. Let's jump into it. Okay, I'm not going to do announcements yet because there's always people who show up late. So let's back it up a little bit. Where we left off. And this will be important because we're going to be doing this in lab today. So again, the last thing we talked about when we talked about these 4 hypotheses was this idea of the law of segregation. That'll definitely be on the exam. Just remember, when we talk about the laws of segregation, the things that are getting segregated are those homologous chromosomes, right? So you learned from the last chapter, when you make sperm or egg, right? When you make these gametes, you know, those homologous chromosomes separate. So if you were to look at any of your gametes, you only have one of each homologous chromosome, unless something went wrong, right? So if you look for chromosome number 2 in any of your gametes, 50% of them will have the mom version, 50% of them will have the dad version. That is the law of segregation. So remember, we're talking about those homologous chromosomes segregating. And also, as you're going to learn later in this chapter, it's not necessarily that you're going to learn that, but we're also going to look at the law of segregation from a different context. So yes, it is the chromosomes, those homologous chromosomes that segregate, but also there's genes on those chromosomes. And since this chapter is all about genetics, that's what we're looking at, is the genes, right? So you're going to learn, or excuse me, once again, you're going to look at it from that perspective. We're going to look at it from that context. We're like, all right, you've got this gene, you've got this allele of this gene that you got from your mom, you got this allele of this gene that you got from your dad, and when you produce gametes, they segregate. That's the law of segregation. And where we left off, let's talk about this, which are Punnett squares. This is what you're going to have to do in lab today, towards the end. But anyway, let's look at this. Some of this stuff you already know. So imagine, again, these are hundreds of fat flowers, if not thousands, but the picture is oversimplifying. But let's say you just take this one purple flower plant and this one white flower plant, right? This thing's going to produce gametes. This thing's going to produce gametes. I don't like this picture because it would be better if it showed that it is actually, because if that one on the left, that one, that purple one is homozygous dominant. So really it should be PP, right? Big P, big P. And the one on the right is homozygous recessive. So really it should be little p, little p. And I know it does say all big P or all little p, and that is true. But I like to see the fact that they're together because this thing hasn't gone through meiosis yet. So yes, they are all big P on this side and they are all little p on this side. But technically those do go through segregation too. Now granted, it doesn't matter that they go through segregation because again, like this picture correctly depicts, no matter what, this purple flower plant, when it comes to that gene, it's only going to offer a big P, right? No matter what, that's all it has to offer. Just like this white flower plant, when it goes through segregation during meiosis, no matter what, the only thing it has to produce is little p's, right? So 100% of the gametes for the purple flower are going to be big P. 100% of the gametes for the little white flower are going to be little p. But anyway, they segregate, then you cross them, which they didn't show, but then here's the offspring, right? So no matter what, this will be the offspring, right? Because when you mix something that has all big P's with something that has all little p's, unless there's some kind of thing that went wrong, then the only thing possible to happen is this flower that has a big P and a little p. And that's heterozygous. And I'll officially introduce you to those terms later. The big P, little p, right? That is heterozygous. It has a dominant big allele and it has a recessive little allele. Now, when that thing goes through meiosis and produces gametes, those, again, they segregate. So therefore, half of the eggs or sperm, whatever it may be, for when we're talking about these gametes, half of them are going to have big P, half of them are going to have little p. You guys with me so far? Does that make sense? That's the only way it can work. That's what it has to offer. It has big P's and little p's. So if you're going to literally get a microscope and look at the sperm or egg of these flowers, it stands to reason statistically, about 50 of them you would find with a big P. Of course, you couldn't see it with a microscope, but let's pretend you could. 50% of them would have a big P, 50% of them would have a little p, right? So then to figure out what would happen if you crossed two of those flowers, and again, this picture doesn't do a great job, but imagine it was like this purple flower crossed with another one of those purple flowers that's also heterozygous, right? And that's what we see here. So again, 50% of the chance, 50% of the time it's going to be a big P, 50% it'll be a small p. For the other plant, it's the same thing. 50% of the time it'll be a big P, 50% of the time it'll be a small p. So then you can use a Punnett square to see the possible outcomes when these things mate, right? So there's a 1/4 chance that you get a big P, big P. Hopefully you can see that, right? And then there's also a 1/4 chance here and a 1/4 chance here, which adds up to a 1/2 chance, right? So there's 1/2 chance, 50% chance that you'll get that homozygous, heterozygous. And then there's a 1/4 chance you'll get little p, little p. And that's what you're going to be doing in lab today. Now, granted, this was, again, heterozygous crossed with a heterozygous. Sometimes you'll have a homozygous dominant crossed with a homozygous dominant. Sometimes you'll have a homozygous recessive crossed with a homozygous dominant, right? All the different combinations is what you'll be doing in lab. This is just the one combination. And when you do this, this is the ratio you get, and this is what you'll be doing in lab as well. The phenotype ratio, there's 3 purple to 1 white. You can see that, right? There's 3 purple flowers to 1 white. One of those purple flowers is homozygous dominant. The other one's heterozygous, and then only one of them, of course, is homozygous recessive. Because remember, if it's dominant, all you need is one dominant allele to show that dominant phenotype, which is why these two here, these heterozygotes, even though they do have a recessive allele, one recessive allele, they're still showing that dominant phenotype. So that's the ratio. And then, of course, you can look at the genotype ratio, like how many genotypes do we have? Well, we have 1 that's big P, big P. We have 2 that's big P, little p. And we have 1 that's little p, little p. So 1 to 2 to 1 ratio. This is what you need to be familiar with. We're going to do it in lab. If I get time, I'm not sure if I have the time, but I would like to make up a bunch of these different hypothetical situations and give them to you as like extra credit homework. You know, like you can do it if you want and apply it to your extra credit, or you just don't have to do it at all if you don't want. But you will have at least a couple of questions like this on the exam. And I will make this, unfortunately, a little bit more complicated later. So you can see this now, figure out the ratios, but later we're also going to talk about the rules of probability and add a little bit of math to this. So instead of just a ratio, we'll also be like, hey, what are the percent? You'll get a question on the exam like, all right, if you mix this thing with that thing, what are the chances of then having this as the offspring? So, and again, we'll cross that bridge when we get there. Any questions about this slide? All right, I also want to point one more thing out, and this is more for your own knowledge as opposed to anything for the exam. This is a very academic conversation. What we're saying here is if you were to cross a bunch of these heterozygous flowers, like thousands, if you will, thousands of them, and then, right, you know, we do this to figure out the probabilities, and again, we'll talk more about probabilities later. What this is really saying, in all actuality, it's not like you cross this purple flower once, and then you're going to come up with 4 offspring, and this will always be what those 4 offspring are. More accurately, and again, this is for your own knowledge, I'm not going to test you on this, I'm not going to get that deep, but more realistically, what this is really saying is, if you were to do this thousands of times, then about 25% of the time, the offspring are going to be homozygous dominant. About 50% of the time, they're going to be heterozygous, and about 25% of the time, they're going to be homozygous recessive. So, you know, again, this isn't just like one plant having 4 offspring. This is just an oversimplification of how it really works. And the probability is what we're looking at. So honestly, you might come up with like, you know, if you were to do this thousands of times, you might come out with like, I don't know, 20% of them instead of 25, maybe you only got 20% homozygous dominant. And maybe you got 55% were heterozygous. And then maybe, you know, the other, I don't know, 15% were homozygous recessive. If that didn't add up to 100, then ignore my morning brain. The point is, that's just a general guideline. For the exam, that is the exact guideline, but in real life, that's just like a prediction, if you will. Anyway, so that's a Punnett square. Now let's see it in writing. Here's everything basically I just said. The Punnett square shows us the 4 possible combinations of gametes, which then obviously shows us 4 possible offspring. And each square represents an equally probable product of fertilization. So again, for the exam, when you're asked to use a Punnett square, we will be like, we won't talk about probabilities, like this is it, right? But remember, in all actuality, this is the probability of this happening. Especially think about humans, right? So what you're gonna do today, one of the things you're gonna do in lab is you and some other classmate are gonna mate, right? So we're gonna look at your genotype for a particular thing, their genotype for a particular thing, then you're gonna do a Punnett square to see what your possible outcomes could be. But it's not like just because you mate with somebody, you're gonna have 4 kids, right? Sometimes you only have 1, sometimes you have 8, whatever it may be. The point is, these are just showing the probabilities. Anyway, any questions about Punnett squares for now? Again, we'll come back to them later in lab. And if I get a chance, I'll give you a worksheet that you can do for extra credit. Matter of fact, I'm going to commit to doing that because I know some of you didn't turn in your pre-lab in time for this week, so therefore you're going to lose a bunch of points. So to help make up for those points, I will make this worksheet. So hey, to make up for your lost points, you can do that and apply them there. Yes, sir.
So about the pre-lab, I completely forgot we had that.
There you go.
If I turned in before the lab today, can I come?
Yes, that's another thing, yeah, so, and I don't blame you for not remembering this, but a while ago I changed it because so many people were screwing up the pre-labs. I got rid of the rule where you can't come if you didn't do the pre-lab. I just, whatever, it's just, you can come, yeah.
If you do it late, like you lose some points.
Yeah, but better than nothing. And again, I'm gonna commit to coming up with something so you guys can make up for your lost points. All right, good question, anything else? And I'll have something else to say about that, but I'll get there in a second. All right, so now let's quickly talk about genetic alleles and homologous chromosomes. So remember earlier I was saying, you know, we're looking, when I talk about the law of segregation, I was talking all about, you know, the chromosomes do this, the homologous chromosomes segregate. And then I said, but in this chapter, we're also going to look at it from the context of genes, right? Because each of those chromosomes have genes on them. So if we're looking at genetics and we're focused on one particular gene, then we would say those genes are segregating due to the law of segregation, right? So the homologous chromosomes are still segregating. In this conversation, we're focused on the genes. So we're going to kind of jump into that a little bit here. And let's do the first word for attendance. And I'm not even going to say it yet. Well, no, yeah, just be prepared. The first word for attendance is coming. How about that? And also, I'll remind you, even though it's been the same thing for a while, at the end of all this, the last word for attendance, the last phrase for attendance is going to be you asking me a question about the material covered in this discussion. Anyway, here's some words you already know, probably. Definitely, if you've already done the pre-lab, you're already familiar with this, but these are terms you definitely need to know. 1, for the exam, because I might ask you. And 2, I'm definitely going to use these terms. I got so many of these questions are going to use the term phenotype and genotype. So phenotype, at least in this context, in this chapter, in this portion of the chapter, is the physical appearance. So again, using this week's lab as an example, your chin is a phenotype, right? Is it smooth or is it dimpled? Your eyes are a phenotype. Are they blue or are they not blue? Your forehead is a phenotype. Does it have a widow's peak or does it not have a widow's peak, right? That is your phenotype. I will point out, and we'll talk about this in more detail later, but for now, this is a good definition, but later we're going to learn that phenotype is actually a little bit more than that. It's not always physical appearance, right? So for example, you could either have sickle cell disease or not have sickle cell disease. You can't see that, but it is a phenotype, and your phenotype is the result of your genotype. So it's your genes, those combinations of whether you have dominant or recessive alleles that make up your phenotype, which again is like what I was saying, even though I'm jumping the gun here. Sometimes your genotype doesn't result in something that you can see. Sometimes it results in something else. But so for now, we can say phenotype is the physical appearance. That's definitely what we're focused on this week's lab. So for now, we'll keep it at that. But just know later, we're going to realize that it goes a little bit deeper than that. Anyway, so when we're talking about what Mendel did, again, these 2 bullet points are just reminding you of what we already said. When he had that F1 generation, and they were all entirely heterozygous, when he crossed them, right, he found out that 1/4 of those F2 offspring had that. Oh man, this is a really dumb bullet point. You know what? Just ignore that because it's missing too much information. And it was just, I should delete that. That was it's just a repeat of stuff we've already talked about, so we don't need to talk about it. I guess basically what I should do is basically bring back what was on this previous slide and say it. Well, we already did talk about it. You know what? This slide should be before this slide, right? Because here I was talking about phenotypes and genotypes. So, yeah, sorry about that, guys. Things get shuffled around. Anyway, first word for attendance. And this is, I'm gonna give you multiple choice here. What would you call the physical appearance? Would it be phenotype or genotype? Physical appearance of something. That is the first word for attendance. All right. Moving forward. Well, first of all, are there any questions about phenotype or genotypes? All right. So the next slide I'm going to show you, and I may have shown you this picture before, I don't remember. The next slide I'm going to show you is a picture, well, it's a drawing of a pair of homologous chromosomes that have some alleles of the same genes. Obviously, that's kind of redundant. Alleles of the same genes. Yeah, remember, an allele is a different version of the gene, so obviously it would be the same gene. And also, now that I think about it, there's also, in this hypothetical situation, there's going to be 3 different genes. So maybe saying the same gene isn't correct. Anyway, there's going to be a pair of homologous chromosomes. You're going to see some alleles on there. And now I'm introducing you to a new term, gene locus. That is the specific location of a gene on a chromosome. You need to know that. So the alleles of a gene reside at the same locus on homologous chromosomes. I have told you that before. I just didn't use the word locus. I used some hypothetical examples when we were first talking about homologous chromosomes. I was saying something like, all right, if you were to look at my chromosomes, and if the gene for hairy back was on the very bottom of chromosome number 17, when you looked at my chromosomes, that means for every other human who doesn't have something wrong with them, they would also have the gene for a hairy back on the very bottom of chromosome number 17. Does that make sense? That's how homologous chromosomes work. Those genes are at the same location on the same chromosome for every species, every individual of that species. Again, sometimes things do go wrong. We've already talked about that. Try to remember, there's like translocations, all these things that can go wrong with meiosis. But generally speaking, that's the truth. But of course, not everybody has hairy back, right? Using that hypothetical example that's not real. But so some people might have a version of that gene that says, all right, smooth back. Some people might have a version of that gene that says, all right, hairy back, right? But anyway, let's look at the picture of what we're saying. There you go. So again, these are the gene loci, right? So we can say gene P, I don't know, I'm going to make this up and say this is homologous chromosomes from number 8. I just made that up. We'll just talk about, because for humans there's 23 pairs. Let's just say this is chromosome number 8. And on chromosome number 8, towards the top here, at this loci, right, this location is where you will find gene P. For every human on Earth that doesn't have something wrong with them, here on chromosome number 8, closer towards the middle than the end, you would find gene P. In this particular location, for this particular human, where we're looking at both of their chromosomes, their homologous chromosomes, the one they got from their mom happens to have a big P version of that gene. The one from their dad also happens to have a big P from that gene. Therefore, for this gene, this person is homozygous dominant. And you definitely, again, these are terms you need to know, right? You need to know homozygous dominant, heterozygous, homozygous recessive. So once again, looking at this chromosome, this homologous chromosomes, and I'm saying it's number 8. If you look right here in the middle of this homologous chromosome, for any human that doesn't have something wrong with them, at this loci, at this location, you will find the gene A, whatever that gene is, right? And this particular person happens to have a little a and a little a for that gene, right? They got a little a from their mom, they got a little a from their dad, therefore they are homozygous recessive. And that's just this human, right? I might be big A. He might be big A, little a. He might be little a, big A, right? So the alleles may differ depending on the human you're looking at. But for us, this location is where you would find that gene. And then finally, same thing here, right? So here, at the very bottom of chromosome number 8, for all humans that don't have something wrong with them, you will find gene B, some hypothetical gene B right there. This particular person happens to be heterozygous for this gene because they got a big B from their mom and a little b from their dad. So they're heterozygous. Does all that make sense? And again, this is all hypothetical. This doesn't have to be chromosome number 8. These genes are all just made up. Doesn't even have to be humans. We could be talking about wombats or kangaroos or alligators, whatever. What confused me when I was a freshman was I was looking at this picture in the textbook, and it said here, Gene Loci, and it would point to it. But then it said also, it would point to it and say alleles. So I'm like, well, what is it? Is it an allele or is it a loci? That confused me, which sounds pretty stupid, and it was like maybe pretty stupid. But so just to be clear, these are alleles, but we're also pointing out that this is their location, right? So this is the location, the loci of this gene. This is the location of this gene. This is the location of this gene. And then you just have different alleles for those genes. Any questions about that slide? Okay. And once again, before I move forward, I'll remind you, when we're talking, this is all genotype, right? And it's not written here. I want to remind you what the phenotype would be. So this person obviously would have the dominant phenotype because big P, right? It's a dominant allele and a dominant allele. This person, or I guess it's the same person, for this gene, they would also have the dominant phenotype because they have a big B and a little b. Now, little b is recessive, but big B is dominant, which means it's dominant over that recessive allele. It doesn't matter that there's a recessive allele. It does have a dominant allele, therefore it's going to show the dominant phenotype. And the only way an individual can show a recessive phenotype would be in this situation, where they have homozygous recessive genotype, right? You have to have two recessive alleles to show a recessive phenotype. Any questions? All right. So again, law of segregation is pretty simple, right? I'm saying homologous chromosomes segregate from each other during meiosis. And then we even change the context a little bit, right? So if we're a geneticist and we're looking at gene P, then when this person goes through meiosis and produces sperm or eggs, this chromosome is going to go one way, this chromosome is going to go the other way, right? They're going to segregate. Therefore, and again, we're just geneticists looking at gene P. We're probably not even talking about the chromosomes segregating, even though they are. What we're talking about, since we're studying gene P, we're like, yeah, the gene P is getting segregated, right? So that part's pretty easy. But I'm about to make it a little bit harder because, in my opinion, these two terms sound somewhat similar. So make sure you understand the difference between the two. We're done talking, well, for now, we're done talking about the law of segregation. Now let's talk about the laws of independent assortment. And you've already been that's what I'm looking for, introduced to this. The law of independent assortment, we introduced you to this when we talked about meiosis. So we'll come back to that. First, let's do reminders. So remind you, exam 2 is closed. A lot of you missed it. I don't know how. I did reminders every day, just like this, where I said, this is the due date for exam 2. This is the due date for exam 2. This is the due date for exam 2. Sometimes when you sit in your attendance words, I would even remind you of right back, hey, you got this many points. Also, don't forget, this is the due date for exam 2. Anyway, it's closed now. Some of you got a 0. But that's okay, sort of. Everyone can retake the exam. But just like the last one, which is not closed, just like the exam 1, go back in there. It's open for you. Take it as many times as you want. It's not timed. It's open book. Once you're satisfied with your grade, email me, and I will average your first grade and your last grade and change your grade. Yes?
You see your grade, mine just says finished submitted.
Oh, that's true. Since I opened it back up, it won't show you your grade. It's a good point. So you can just email me and I'll tell you what you got. Actually, if you click on it too, like it won't show you your grade, but if you, well, no, it won't do that either. That's a good point. Yeah, I'll try to change the settings for that too. But yeah, until I change the settings, you can email me and I'll give you your grade. Great question. Anyway, make sure you email me when you're done. I will average your first grade and your highest grade. So for some of you, your first grade is 0. If you get 100%, you'll at least get 50 points. This closes when exam 3 closes. So you should see a pattern here, right? Any questions about exam 2? Any more questions? That was a good question. So again, I'm going to try to tinker with the settings so you can actually see the grade that you got. But until then, email me if you want, and I'll give you your grade. But you know what? Give me a day first, please, because I want to tinker with those settings. Obviously, I don't want to be emailing everybody back their grade. I'd rather you just be able to see it yourself. So give me a day to figure out those settings and fix that for you. Another thing is lab is today at 10 as usual. We will be meeting here. It's not virtual. Doors are going to lock at 10:01 a.m. If you're not here, you don't get credit. So make sure you're on time. The lab report is now past due. I'll just remind you what the syllabus says and what I've been saying for a while. Officially, I don't accept late work. Unofficially, I'm telling you, turn it in. I prioritize new work, but when I'm caught up with new work, then I always go back and grade late work in the order in which it was received. So the sooner you turn it in late, the better. What else? Next week's lab is, yes, question?
For the lab report, like, it says here that it was due on like probably 3 days or 2 days.
It said, what's it say, when does it say it was due? Sorry.
It says due on Friday on the.
This Friday?
That's what it says.
Okay, never mind then, I'm sticking with that.
Yeah.
All right, that was a mistake. It shouldn't have said that. But if that's what it says, that's what it says. Thank you for letting me know. All right. I'm not changing it if it was already there. In my opinion, originally I was saying it should have been due last Wednesday because that's 2 weeks after the exam. I mean, two weeks after the lab. But I was like, I don't want to make it due during spring break. So I'm going to change it to Monday. But hey, if that's what it says, that's good. So hey, your lab report's due this Friday. Is that right? You said Friday?
Yeah.
Okay, there it is. All right, your lab report's due this Friday. I remind you, everything you need for that lab report is under the announcements. If you go and look at the announcements, there's one for photosynthesis lab. It's got that PowerPoint that shows you how to write a lab report. It's got the video of me telling you how to write a lab report. It has the data that you need. It has all the links that you need. Everything's there. And also, I'm here to help you. You don't have to do it alone. I'm glad to help. Anyway, yeah, next week's lab. It is 100% virtual. Technically, there's like 3 questions of a pre-lab, I think. I'm not even worried about the pre-lab. It's all virtual. We will still meet in person. However, if you do get it done and turned in before next week, I might say like, hey, never mind, you're good. You don't have to come in. Unless you hear that from me though, plan to be here next week. Independent works, now that I've submitted midterm grades, I'm now eventually here once I get a moment. We'll add your independent work points to Moodle. I've been mentioning this before. For almost all of you, that'll mean your grade will drop. But keep in mind, it's Moodle. Everything's unofficial. It's not official until the end. So I only wanted it to drop now so you don't get that surprise at the end. I don't want you to think you have a B and then end up with a C and only 2 days left to do something about it. Actually, no, by then it'll be 0 days. Anyway, like I said last time, the last day to drop this course is sometime this month. I forgot exactly when. Some of you should highly consider it. And for those of you who should highly consider it but decide not to drop, you should really let me help you. Let me get you caught up. I'm here to help. And then finally, I'll remind you, the Friday before Easter and the Monday after Easter, everything will be 100% online. Classes, or excuse me, lecture and office hours. Any questions about reminders? Okay, so the next word for attendance is, let's see. The exam 2 retake is now, like I've reopened exam 2 for a retake. When does it close? So exam 2 is now reopened, so you can retake it. When does exam 2, this retake, close? And also, while I'm giving you time to answer that, I'll remind you, now that the exam 1 retake is closed and behind us, feel free to meet me online so I can give you all the correct answers to that exam, give you feedback, and give you a 5% boost to that grade for exam 1. And the same thing will happen for exam 2 once the retake is closed. All right, anyway, let's move forward. Talk about a dihybrid cross. So far, everything we talked about these phenotype ratios and genotype ratios, and if this thing mates with this, the purple flower plant mates with a white flower plant, what are the possible offspring? That was a monohybrid cross, right? Because we were looking at one character. In that particular example, the character was flower color. And now we're going to talk about something called a dihybrid cross. And this is a mating of parental varieties differing in 2 characters. Nice and easy, right? It's in the name. But this is where we start getting into this idea of independent assortment, as you can see up top. So let's talk about that for a second. So let's talk about a hypothetical. If genes for 2 different characters were inherited together, Well, let's talk about what happened to that first of all. And what I mean by that is, say for example, chin dimple and eye color, right? Let's say for some reason, and this is not the case, but let's say they were both on chromosome number 8, right in the middle, right next to each other. And that means if you got your mom's gene, or excuse me, allele, her version, the chin dimple gene, then you would also get your mom's version of the eye color gene, because they'd be right next to each other, right? If they were inherited together. In other words, if they were inherited together, and all this fancy words basically just says, if they were inherited together, you would just see the same ratio like we saw with that monohybrid cross that I already introduced you to, right? So if we did a heterozygous cross with a heterozygous, then this is, we would get a 3-to-1 phenotypic ratio like I already showed you, if the genes were inherited together. But that's almost always not the case. They're almost always sorted independently. And I don't know how to say this without getting it too complicated. Let's just say it like that. This will make more sense when I show you the next slide, but let's just put it this way. When you see a 9-to-3 to 3-to-1 ratio, you should associate that with a dihybrid cross. That'll just be my one thing, because this is a 100 level course. I'm not getting too much into it. The next slide is going to make it help a little bit more sense. But here we go. I get to show you. So the situation on the left is the hypothetical. It doesn't happen, right? It doesn't almost never happen. So what this is saying is if the gene for P color also migrated or was associated with a gene for P shape, right, then this is what you would end up with, right? So here we have a heterozygous for both of them, right? So they're big R, little r for the color. They're big Y, little y for the shape. And in this case, yellow is dominant, green is recessive. And what was the other part? Oh yeah, the wrinkliness would be recessive, and being smooth would be dominant, right? So if these things, like if they were together, then when this thing goes through meiosis and goes through, those things segregate, then you would always have big R going with big Y, right? And you would always have little r going with little y. And you would end up with, again, with this 3-to-1 ratio. But that's not what happens. What actually happens is these things segregate independently of each other, right? So when this thing produces gametes, it's going to produce some with big R, some with big Y, some with little r, some with little y, and all these different combinations, right? So it could be big R, big Y, it could be little r, little y. It could be little r, big Y, or big R, little y. Does that make sense? I'm hoping you guys see what I'm saying here. When that thing goes through meiosis and all those things segregate, then that's the different possibilities. So again, if we're looking at two different characters, if you were to look at the gametes, if you could somehow look at their sperm or egg, then you would find 25% of their sperm have big R, or egg, have a big R and a little y, big R, big Y. 25% of their sperm would have a little r and a big Y. 25% of their sperm would have a big R and a little y. And 25% of their sperm would have little r, little y. So basically, for this 100 level class, what I'm really saying here is that things get more complicated when you're looking at more than one character, right? And again, you should associate this phenotypic ratio of 9-to-3 to 3-to-1 with a dihybrid cross. I know it seems confusing, and I will say this, don't let it confuse you too much. This 100 level course is not that important. So once I'm done teaching you this and maybe giving you a couple of questions about it on the exam, putting it behind us. This concept will not come back. So if you don't truly understand this concept, it's not going to hinder you from learning later concepts. And to me, that's the most important, right? Is this building knowledge. So sometimes you have to know older stuff to learn new stuff. This is not one of those things. So don't let it stress you out. But anyway, again, what we're trying to demonstrate here is independent assortment. So let's talk more about it. The dihybrid cross that I just showed you supports this hypothesis. I'll have a little star next to that. But yeah, it's not really a hypothesis anymore. Remember, big picture here, I'm trying to tell you what Gregor Mendel did when he was doing all this research. So at that point, it was a hypothesis. Now we just know this is the way it is. Anyway, that dihybrid cross supported his hypothesis that each pair of alleles segregates independently of each other during the gamete formation. Another way of saying that is the inheritance of one character has no effect on the inheritance of the other. And this is Mendel's law of independent assortment. Independent assortment is seen well, yeah, that's it, right? So this is the law of independent assortment. The inheritance of one character has no effect on the inheritance of another. And in my opinion, to me, this makes a lot more sense when you think about the way I taught it in the last chapter, when we looked at it from the other point of view. The other way we looked at it was, I was saying, I'm not drawing it, I'm doing it like this. When the chromosomes line up in the middle of the cell for meiosis, it's not like it's all random how they would do it, right? So if this thing had 3 chromosomes, it might have chromosome number 1, right? It might be from the mom. Chromosome number 2 might be from the dad. Chromosome number 3 might be mom, right? It could line up that way, or it could line up all mom on one side, all dad on the other, right? Again, picture those are chromosomes and not letters. That's all independent assortment is saying. And remember, I gave you some sort of number, like the chances of that one particular sperm that made you, fertilizing that one particular egg that made you, was like 1 in 64 trillion. I don't know if you remember that. And that's all based on the way those homologous chromosomes line up in metaphase 1, right? So it's not all mom on one side and all dad on the other. For all 23 pairs of your chromosomes, it could be any way, right? They all line up independently. And of course, each of those chromosomes has genes on it, which brings us back to the context of this conversation, right? Because we're looking at the genes as opposed to the whole chromosome. Any questions about that slide? The next slide I'm going to show you is just a visualization, just another example of this 9 to 3 to 3 to 1 ratio. This is from the old textbook. I'm not going to test you on it, it's just an example. So here we have a black dog. All right, first of all, let me tell you this. In this situation, being black is dominant, being brown is recessive. Being able to see is dominant, being blind is recessive. So here we have a black dog that can see. We don't know its genotype, but we know that at the very least it has one big B, because it has the dominant phenotype, and we know that it has at least one big N, because it has a dominant phenotype. Here's a black blind dog, so we don't know its genotype for its fur color, because all we know is it has at least one big B, right? Because it's showing the dominant phenotype. However, since it's blind, we do know the genotype for that, right? It has to be homozygous recessive, little n, little n. Then, of course, on the next to that, we have a brown dog who can see. So again, if it's brown, that's recessive phenotype. Therefore, we do know its genotype, which is little b, little b. It has to be to have that recessive phenotype. However, it can see, which again is a dominant phenotype, so we don't know the genotype. All we know is it's one big N. The other one, it could be big N, or big N, little n. Doesn't matter. It's going to show that dominant phenotype. And of course, at the very right, we have a blind brown dog, which means it's completely recessive, right? For both characters, it's recessive. So little b, little n, little n. Now, if you were to get 2 heterozygote dogs and mate them, this will be the results, right? You could expect 9 of them to be black and that can see. You would expect 3 of them that can be blind that are black. You would expect 3 of them to be brown but can see. And you would expect only 1 of them to have both recessive traits, which would be brown and blind. This is just an example. I'm not going to ask you any questions about it. It's also not even really any new information. This is the same thing I was showing you with the pea plants. I'm just not showing you the Punnett square. It's the same basic information. The 9 to 3 to 3 to 1 ratio is what you would expect. All right. Any questions about that? All right. So speaking of this, right, like I said, for this dog, we don't know its genotype at all. For this dog, we don't know the coat genotype. For this dog, we don't know the vision genotype, right? So the next thing I'm going to teach you is how we can use test crosses to determine genotypes. So first of all, you need to know what a test cross is. Test cross is a mating between individual of a dominant phenotype but an unknown genotype with an individual that is homozygous recessive. In other words, less complicated way of saying it is, a test cross is when you're trying to figure out a genotype So you're crossing two individuals, one of which has the dominant phenotype, and we don't know the genotype, and the other one has the recessive phenotype, therefore, obviously, is homozygous recessive. But here's how that works. So here we have, again, this black dog. Now we're not even focused on vision. We're only looking at the color of it. This dog is black, which is the dominant phenotype. Therefore, we know it has at least one big B, right? We don't know what the other one is. Is that big B or little b? We don't know. As long as it has that one dominant genotype, one dominant allele, it's going to show the dominant phenotype. This dog, on the other hand, it's got the recessive phenotype. So we do know its genotype. We do know it has to be homozygous dominant. So here's what we can do. We could cross these two dogs and look at their offspring. If, which one do I want to start with? Yeah, okay. Let's start with this one on the right. If you were to cross these dogs and any of them come out brown, any of the puppies come out brown, you would know that black dog's genotype would have to be big B, little b. Does that make sense? Because if it's brown, if the offspring is brown, remember, a brown, like it says right here, is little b, little b. So if it's brown, that means it got a little b from its mom and a little b from its dad. Both of the parents had to have at least one little b to give, right? So if you cross those dogs, and again, you would expect a 50-50 ratio, so you would expect half of the puppies to be brown, half of the puppies to be black. And again, this is all probability. Might not always turn out that way. But for sure, if you had a brown dog, you know that parent dog right here would be heterozygous. Now, if this dog was homozygous dominant, as you can see on the left, then 100% always the dogs would be black. Does that make sense? Because this dog only has a big B to offer, or excuse me, only has two big Bs to offer. So it doesn't matter, it's always gonna be heterozygous. It'll always be black. The only reason I don't like this example is, again, if you were doing this in real life and you were crossing these two dogs and all the puppies were black, you could guess, you could say, all right, well, all the puppies are black. So surely this thing must be homozygous dominant. But again, for the sake of what you need to know for the exam, what I told you was more important. Now what I'm about to tell you is just for the sake of your own knowledge. Think about that in real life. This is all probability. So it is technically possible that in this situation, with if it was heterozygous, it is technically possible that all the offspring would be black. You would expect to have at least one brown dog, but it's all probability. It's all a flip of the coin. So technically, that could also end up in 100% black dogs. I guess my point is, if you were doing this in real life, the only thing you would know for sure is if it had a brown dog, that this dog is homozygous, heterozygous. Because yes, technically, by the rules of probability, if they're all black, that still doesn't necessarily tell us anything. Now, obviously, the more black dogs it has, the higher the probability that this dog is homozygous dominant. But it's all just probability. Which brings us to the rules of probability. And this is, again, you're going to have some basic math in this exam, none of which you're going to need a calculator for. Of course, I'm not going to say you're not allowed to use a calculator, you are, but it's going to be very basic rules of probability, so let's talk about it. So the genetic crosses that we've been talking about, all these, these test crosses or these Punnett squares for the monohybrid cross, even the dihybrid cross, they all obey the rules of probability. And these rules of probability are the same that apply when you toss a dice, roll dice, toss a coin, draw cards, anything like that. So the first thing you need to know about is the rule of multiplication. You're going to need to use this at least once, maybe twice on the exam, which states that the probability of compound events is the product of separate probabilities of independent events, which sounds complicated, but it's not. So let me ask you this. If I were to flip a coin right now, what are the chances of it landing on the heads? 50-50, right? You could also express that as a 1 and 2, right? A 1.5 chance. Come on. So, what are the chances of me flipping it again and landing on heads?
25.
Yes and no, but yeah, just that, for that one individual event, it's still just 1 and 2, right? 50-50 chance. So, that means then the chance of me doing it twice, like you were saying, I think, and it landing on heads both times, would just be, you multiply the two probabilities and you get 1 and 4. So like he said, the 25% chance. So all you're doing is multiplying. And I'll give you a real life example before I even go to the next screen. What are the chances of, so everything being equal, what are the chances of me and my wife and I having a child next year and it being a male as opposed to a female? 1 and 2, right? So 50-50 again. Now, what if there's some disease that we carry and, I don't know, for some reason, I don't know, there's a 1 in 8 chance of our offspring getting that disease. So on the exam, you might get a question like that, like, there's a 1 in 8 chance of passing on this disease. What are the chances of these two people mating and having a girl or a boy with that disease? And again, you would just multiply the probabilities. In that case, it'd be 1 in 16. Very basic math. Make sense? All right, and here is a visualization of what I'm saying. Right, so when you're looking at, here's a heterozygous female mating with a heterozygous male. When this female, this female went through meiosis and produced a bunch of eggs. So let's say we could somehow see inside of her eggs, every single one of her eggs, or like actually see those of the big B or little b. What are the chances of choosing just one of her eggs out of her entire batch of eggs? What are the chances of getting one of her eggs and getting one with a big B? It's 1 and 2. Right? What are the chances of us getting just one of those eggs and getting a little b? 1 and 2. What are the chances with same with the sperm, right? Same with this male. What are the chances if we could somehow like reach in and just grab a sperm and look at it? What are the chances that we would find one, we just pulled one randomly and it had a big B? The chances would be 1 and 2. What are the chances of doing that and finding one with a little b? 1 and 2, right? So then you talk about fertilization instead. Like, so what are the chances that one of these eggs meets with one of those sperms? And that's all that is, right? So the chances of this is 1 and 4, right? Because 1/2 times 1/2 is 1 and 4. They are each 1 and 4. Does that make sense? If you're just saying, what are the chances of being this egg? What are the chances of it being that sperm? 1.5, 1.5. Multiply the two, 1/4. Everybody good with that? All right. Then the other thing you have to look at to make it a little bit more complicated is then you can add it. I don't even know if I want to get this deep. But look, you can see here, I'll just say it a little bit, I'll just brush on it. You can see here there's a 1 in 4 chance of getting big B, little b. But there's also a 1 in 4 chance of getting big B, little b here. So technically, if you wanted to ask, if I were to ask you, what are the chances of getting big B or big B, little b? You would actually have to add those two because those are the probability of this or this as opposed to this and this. So 1/4 plus 1/4. Do it like that. Yeah. So 2/8, which is also the same as, now that, I'm gonna set up. Anyway, sorry about that. The chances of this happening are 1 and 2, right? Or 2 and 4, I'm sorry. Good grief. It's been a while since I've done that. Anyway, again, you might get some easy question on the exam where I say, what are the chances of this happening and this happening? And if it's the two things, this and this, then you're gonna multiply them. All right, any questions about that? Okay. Next thing we're gonna talk about when we get back will be family pedigrees. And if I have time too, that's another thing. Maybe I can make some family pedigree worksheets that you can do for extra credit. Anyway, family pedigrees, what we'll do when we get back. My last word for attendance is person in the top left has what color hair? Can you see it? Okay, here's an easier one. How many different pictures are on this picture? Don't answer it out loud, but instead, your last word for attendance, other than asking me a question, is how many different pictures are on this slide? And it's not a trick question. All right, so I will see you guys at 10 o'clock. I'm gonna go down to the office to do office hours for a little bit. And if you're online, don't forget you need to send those words before 9 a.m.